So we've said that once you increase in height, you have grown. Once there's a change in your weight, you have as well grown. Once there's change in volume or the amount of space you can occupy, then that is also growth. Then as well, we can decide to use length to determine whether an organism has grown or not. Now, for example, if we take length into consideration and we want to use it to determine whether an organism has grown or not, we are going to actually have that length over a specified period getting plotted against time. So for example, if I come here and have this as my graph, and this graph has two very important aspects to consider. Number one, I have actually put here my growth parameter. For example, I've been measuring the change in height of an organism over a long period of time. So I have here a growth parameter like height. Then I have here the measurement of the change in height with the change in time. I'm going to have this type of curve. Look at that. So that type of curve is actually referred to as the S-shaped graph. And this S-shaped graph has a specific name called the sigmoid curve. I never bothered taking all these parameters like height, weight, volume, and length. I only captured one like length, or I only captured one like height. Then I actually monitored the change in height of this organism with the change in time. I got values. I have plotted the values of height against values of time. I've actually come up with what we call an S-shaped graph. And this graph is called biologically the sigmoid curve. Now, what does that tell you? If you look keenly at this graph, let's try to just describe it. I'll be able to actually realize that a graph of this type, for example, having height maybe in meters or centimeters, and then let's use centimeters, and then you have time in seconds, will tend to have something like that. And descriptively, you can actually be able to realize that the pattern is due to the fact that growth tends to be slow at the beginning. Growth tends to be slow at the beginning or at the first point of growth, and then it speeds up until it later on slows down when someone has aged, or the organism has actually aged. So there's actually a clear picture that someone can actually get from this graph. I repeat, growth first slows down at the beginning, then it quickly increases, and then finally slows down when the curve levels off, or when the organism reaches an adult size. Now, we'll realize that the sigmoid curve can be divided into four very important parts. We'll have this part. Yes, you can be able to clearly see that. Then we will have this part. We will have this part. So we have part A, where growth is slowly increasing, where the growth speedily increases, where the growth slowly begins to slow down, and lastly, when growth has almost stopped. So we have four very important parts in that. So you can be able to have this A, have this as B, have this as C, and lastly have this as D. So four very important parts of that sigmoid or S-shaped curve that we want to descriptively be able to use to explain how growth occurs. Now, if we begin with the first part is called the lag phase. The lag phase. Lag phase where there is slow growth. Lag phase where there's slow growth. Now, at this initial phase, I'll just give a summary so you can be able to follow and be able to write down the short notes. So in this first phase, called the lag phase or the slow growth phase, there's always slow growth. There's always little growth. The growth is slow. Why is growth slow? Because if you look at the curve tends to slightly increase. So that slight increase in the graph tells you that the growth experienced here is very slow. Why would actually the growth be here of a slow rate? Why do we end up having a slow growth rate at this point? It is because one, the number of cells dividing at this point are very few. So number of cells dividing are very few. The number of cells dividing at this point are very few. Is when the organism has begun growing. Is when the organism has begun increasing in size and height, or weight, or volume. That means the number of cells dividing are very, very, very few. Number two, the cells have not adjusted. Cells have not adjusted to the changes in the surrounding. You know, when cells are not well adjusted, when the cells are not well adapted to the surrounding, their growth tends to be slow. Their rate of cell division tends to be slow. They are affected by the environmental conditions that prevail at that moment. So that means at this lagging behind, that means very slow. You see, so lag phase or slow growth phase is where the rate of growth is very slow. There's little growth occurring at that point because we have very few cells dividing at that point because the organism is when it has actually begun growing. So when the organism begins growing, that means the number of cells dividing at that point for the organism to grow are very few. And on top of that, cells have not adjusted to the surrounding in the environment or to the environment. Now, part B, we have what you call log phase. Log phase, I think if you remember, there's a topic in mathematics called logarithm. That means there's a progressive increase, an arithmetic increase, a sequential increase in the number of cells. So log or exponential. Log or exponential phase. So this is the second phase of growth. And during this second phase of growth, the growth is rapid. So there's rapid growth, or the growth will proceed logarithmically or exponentially. So proceed exponentially. So at this point, you can look at this curve. It has drastically increased. It has drastically risen to tell you that the growth is exponential or the growth is logarithmic. So growth rate increases. There's a rapid or increased growth rate. You see, at this point, you've said the growth rate is rapid, then the growth here is at its maximum. The growth is at its maximum. You see? And the rate of growth, you see, the rate of growth is proportionally to the amount of material or number of cells of the organism already present. You see? So rate of growth is proportional, we can say. The rate of growth is proportional to the number of cells present. The number of cells present. Let me take you back to this point. The cells began dividing here, growth began here. So obviously, as the growth proceeds to the second phase, the number of cells must have actually increased. So when the number of cells increase, then that means there's rapid or increased growth rate. The growth proceeds exponentially. There's maximum growth. The rate of growth is proportional to the number of cells present. And because the number of cells present is higher, that means the growth rate must also be higher. And we shall have to look at some of the factors that can actually make this growth rate to be rapid or maximum. Number one, we've said number of cells have increased. Number of cells has actually increased. So when the number of cells has increased, then that means the rate of growth must also increase. Number of cells increased geometrically. Number of cells increased geometrically to mean 2, 4, 8, 16, 32, 64. So you proceed. I think you remember geometric progression in mathematics. That means there's what we call a common ratio. So the number of cells has increased geometrically. That means when the cells increase in numbers, the growth rate will be rapid because we shall have more cells dividing. Now, number two, cells have adjusted. Cells have adjusted to the surrounding. Cells have adjusted to the surrounding. The cells have adjusted to the surrounding. I actually understand when cells adjust to the surrounding, then their rate of cell division must also increase. And that brings about rapid cell division or a higher or greater rate of growth. So cells have increased in numbers. Number two, cells have adjusted to the surrounding. Number three, you see, when cells have to end up dividing, then the environment must be favorable. So, for example, food and other factors such as temperature, you see, and pH are not limiting. Including oxygen. If a cell has to divide, there must be nutrients. Now that is food and oxygen. When nutrients such as food, we're actually discussing here like glucose, and oxygen are present, then the food such as the glucose we're actually discussing gets broken down through respiration in the presence of oxygen. And when the food gets broken down in the presence of oxygen through respiration, then there's energy for the cell to divide. Because cells divide in the presence of energy. Cells can divide when there's energy. So when there's energy, the cells divide. For example, the glucose gets broken down. That is now the food in the presence of the oxygen gas through what we call aerobic respiration. When there's energy, the cells divide. So when cells divide, then that means there's growth. Temperature will be able to activate the enzymes that control the rate of respiration. Look at that. And pH must be favorable for some enzymes to work well. So that tells you that when food is available, oxygen is available, temperature is conducive for optimum, and pH is conducive for optimum or appropriate, then that means the enzymes will speed up the rate of respiration. Glucose will be broken down, that is food, in the presence of oxygen to produce what? Energy. The cells divide rapidly, and there's rapid cell growth or rapid cell division. Then last, you'll also note that number of cells dividing is higher than the rate of cell death. What does that mean? The number of cells dividing is greater than the number of cells dying. So if the number of cells resulting from this cell division is bigger than those dying, the resultant outcome is growth. I guess if we have more cells dividing and only few dying, then the organism obviously ends up increasing in size. Let's say per cell division we have 20 cells resulting. Okay? And of these 20 cells, only 5 cells die. Look at that. That means I'll still increase in size. resultant output is increase in size, increase in weight, increase in mass, increase in volume, and of course, that tells you that the organism has actually grown. So that is the log phase or the exponential stage. So one, increase in cells. Geometrically, cells adjust to the surrounding so that they can actually be able to divide and increase in numbers. Then food, oxygen, temperature, pH are not limiting factors to the cells. So they have a conducive environment for them to divide. Then lastly, the number of cells resulting from the growth is higher than the number of cells dying. That tells you the organism ends up increasing in size, and that is growth. Now, we'll turn back to the third phase of this, and the third phase is called decelerating. Look at that. We can be able to notice that the rate of growth has begun decreasing. Look at that. The rate of growth has begun decreasing before it even flattens out. Look at that. So this third stage is called decelerating. So decelerating phase. Decelerating phase. So this is the third phase during which with time growth becomes limited. Growth becomes limited. That means growth has begun slowing down. Growth has begun slowing down. It had drastically increased, but now it has begun slowing down. Why? We shall give the reason behind that. We need to have some factors that can actually be able to act as our evidence to confirm that literally growth has begun slowing down. Growth has begun slowing down. And we shall actually do this. We shall actually be able to notice that one, maybe most cells are fully differentiated. Cells fully differentiated. Remember the fact that the decelerating phase has to involve cells ending up with the time becoming limited in growth. And when cells become limited in growth, so this is what we call limited growth. And this limited growth is brought about by internal factors and external factors. So this is called decelerating phase. There is limited growth with time. Growth has become limited. Growth becomes limited because of internal and external factors. What are some of these internal and external factors that can actually bring about limited growth? One, maybe cells have fully differentiated. You see, when a cell divides, it will give rise to 2 cells, then later on 4 cells, then later on 8 cells. Geometrically, we shall end up having millions of cells in the body of this organism. Those cells will then become specialized or differentiated to become, for example, in animals, sperm cell, kidney cell, liver cells, and other cells, even the skeletal muscle cells. In plants, we can have what we call the xylem, we have what we call the tracheid, we have what we call the sclerenchyma, the parenchyma, the collenchyma, the epidermis, the root hair cells, we have what we call the palisade cell, the spongy mesophyll cells, the guard cells. All these cells have become differentiated. And once a cell becomes differentiated, it cannot be able to further divide. It has actually become specialized to perform a specific function. So it cannot be able to again end up dividing. So when the number of cells resulting from this cell division become differentiated, you see, you shall not have any other cells dividing because all of them have become differentiated. And that means there will be no any further increase in the number of cells. And that means the growth rate becomes slow. Look at that. So cells began dividing, they gave a rise to a number of cells, and these cells now became differentiated. We shall not have any other cells to divide. So the growth rate slows down. So if the cells become fully differentiated or specialized to perform specific functions, they may not end up dividing further. That slows down growth. Number 2, we shall talk about fewer cells dividing. Fewer cells still dividing. Fewer cells still dividing. Why? Because I've said, for example, a good number of cells have become differentiated. So, if a good number of them become differentiated, we shall have fewer cells remaining, still dividing. And if fewer cells still remaining, dividing, that means we will actually have what you call a slow growth rate. Okay, number 3 that you need to know, we have what you call environmental factors. The environment may not be so favorable. So environmental factors can make the growth rate to slow down or to become limiting. And some of these factors can either be internal or be external. So some of these factors can either be internal or external. For example, shortage of oxygen and nutrients. Shortage of oxygen and nutrients can be a limiting factor. You see, remember, we had the log or exponential phase where there was a rapid cell division. The cells drastically increased in numbers. So the number of cells can be now more than the nutrients available and oxygen. So if we have more cells and a small amount of nutrients and oxygen, some cells may lack oxygen and nutrients. Will they divide? No. So that means that can be a limiting factor to bring about slow growth or a decrease in growth or a decelerating rate of growth. Look at that. So that means decelerating means there's a decrease. Decelerating in terms of motion, there's a decrease in growth. So it's because of maybe the shortage of oxygen and nutrients. You see, when oxygen and nutrients become limited, then that means growth also becomes limited. So shortage of nutrients and oxygen because of the increased number of cells can make growth to become limited. Then 2, space can be limited. So space is limited. Why? You remember, we had this space. The cells became 3. Remember, the cells are going to dividing. You see, when the cells increase in numbers, the space within the environment, in as far as the cell division is concerned, may become limited. So if the space becomes limited, that means the cells won't have enough space for them to further divide. That can become a limiting factor in terms of growth. So limited space can be a factor. Limited space can be a factor. So we have said this decelerating phase can have limited growth or slow growth or a decreased rate of growth because of 1, cells becoming fully differentiated. Look at that. Cells becoming fully differentiated. So we do not have any other cells to divide because they are now specialized. They have left actually that phase of dividing and they are now performing different functions. Then you have fewer cells still dividing because a good number of them have either reached their maximum size, have differentiated, or some are dying. So you only have few cells now still dividing. That can bring about slow growth. Number 3, environmental factors like these factors becoming internal or external, such as shortage of oxygen and nutrients because of the increased number of cells. Number 2, space becoming limited. Or you can talk about accumulation of wastes. Accumulation of wastes. Remember, I told you that in a cell, there's nutrients like glucose. The nutrients undergo respiration, specifically aerobic respiration, where, for example, glucose can get broken down to produce energy, you see, in the presence of oxygen, so that the cell can divide. So when there is breakdown of the glucose to produce energy, so that the cell can divide if it has energy, then that means the waste can accumulate. Remember, when you talked about respiration, you said we break down glucose, produce energy, carbon dioxide, and water. So carbon dioxide can be a waste product. So when this waste product or waste materials accumulate in the cells become poisoned. You see, the wastes become harmful to the cells. Then that means the cells cannot further be able to divide. So when the wastes accumulate in the cells cannot divide. And that means there will be slow growth. The growth rate becomes slow. Look at that. So accumulation of metabolic wastes or metabolic waste product can limit growth or can inhibit growth. You see? Now, sometimes limited resources can slow down growth. So limited resources such as carbon dioxide. Remember, In plants, we need carbon dioxide during photosynthesis so that you can actually manufacture food. When carbon dioxide becomes limited in plants, that means there will be no photosynthesis. At the end of the day, plants won't make their own food. And if food becomes a limiting factor, then that means there will be no growth, because these cells in the plant are relying on the food the plant can make for them to actually divide and increase in numbers to bring about growth in plants. So if CO2 or carbon dioxide from the atmosphere becomes limited, that means there will be no photosynthesis in the plants. The plants will not make food, and in the absence of food in form of glucose, there will be no respiration, there will be no energy, cells won't have energy, and there will be no growth. So limited resources such as carbon dioxide can actually slow down growth. And the last phase of that graph is called the plateau. The plateau phase or the stationary phase. Plateau or stationary, you can also call it stationary. So the plateau phase or stationary phase. I think if you talk about plateau in geography, it clearly communicates what you're supposed to know. And stationary means growth has become constant. So this is where overall growth has ceased. So you can talk about growth ceasing. Growth has ceased. Growth has stopped. Growth has ceased or growth has stopped. And here, if you are considering height or weight, all these parameters have become constant. Parameters have remained constant. Parameters remain constant. So there's no any increase in size, no any further increase in height, no any further increase in weight, no any further increase in volume. Growth has remained constant. So plateau phase or stationary phase is where growth has ceased or growth has stopped. And that means parameters we had put into consideration like height or weight have remained constant. What could be the reason behind this? One, maybe rate of cell division. Rate of cell division is proportional to rate of cell death. Rate of cell division is proportional to the rate of cell death. Imagine you have 10 cells resulting from a particular cell division. Then all those 10 cells die. What does that mean? Will there be any change in height or weight or size? No. So if the rate of cell death is equal to the rate of cell division, that means there will be no any further increase in the number of cells. There will be no growth. Growth ceases. Growth stops. Growth becomes constant. The organism remains at its particular point in terms of height or weight or size. And at the end of the day, there will be no any further increase in the size of this organism. So growth will actually be able to have actually ceased or stopped. So plateau or stationary phase is where growth has ceased. Overall growth has actually ceased. There will be no any further increase in size, no any further increase in the number of cells, because the rate of cell division is equal to the rate of cell death. The number of cells resulting from a particular cell division is equal to the number of cells resulting from a particular process in terms of cell division. And that means cells resulting from the cell division are ending up all of them again dying. So there's no an overall increase in the number of cells, no overall growth, no overall increase in size and mass or weight. So that means cells have ended up not increasing in number. Now, nearly the last aspect to consider, all cells have fully differentiated. All cells have fully differentiated. All cells have fully differentiated. So when nearly all cells are fully differentiated, then that means there will be no any further increase in the number of cells. Yeah. So nearly all cells have fully differentiated to mean that all the cells have ended up specializing to perform specific functions. So they are not further dividing. If there's no any further division of the cells, then there's no increase in size and mass. There's no growth. Growth has actually stopped or ceased. That's what we call the plateau, the stationary phase. Now, you see, sometimes we might have this type of curve called the sigmoid or the S-shaped curve. But the curve that can result from any representation of growth will depend on the parameter that actually gets put into consideration. So if you're using height and someone else is using weight or mass, then that means the curve will differ. It will slightly differ. So the curve differs. Curves differ because of one, the parameter you're considering. Parameter considered. Are you using height? Are you using weight? Are you using mass? Are you using volume? The parameter won't be the same, and that means the graph can also be slightly different. Now, number two, the species you're actually considering might also be a factor to actually give us a different shape of the curve. I don't think the rate of growth in plants and the rate of growth in animals might be the same, or even the rate of growth in different animals. The rate of growth in different animals might differ. So the species you're considering in terms of growth can make the graph to be slightly different. Look at that. You see? So the graph can differ, or the shape of the graph can differ because of the species under consideration. The species under consideration can actually bring about a difference in the graph. And lastly, internal factors. There are those organisms where, for example, internal factors can be limiting. Just look at a scenario whereby an animal cannot live without food like a human being. You are eating every day, every time, every minute. That means the supply of nutrients to your cells is continuous. You see, but now remember, you can have an example of a plant where carbon dioxide can be limiting. So the plant may not have enough carbon dioxide. If there's not enough carbon dioxide, that means the plant may not end up photosynthesizing. It will end up not having nutrients in its structure. So cells may not have enough nutrients or may not have nutrients for growth. So internal factors in the organism can make the organism to end up having a different shape of the curve in terms of its growth. If an organism has an adequate supply of nutrients, such as glucose and oxygen, and there's an appropriate pH and temperature, that means the rate of cell division will be very, very high. Look at a scenario whereby a plant ends up having a limited supply of some internal factors, and it ends up not having a normal rate of cell division and cell growth. The curve getting obtained from that plant's growth and that of an animal with an adequate supply of nutrients will be different. You see, so that means internal factors can make a curve to be different depending on what you're looking at in terms of growth in the plants. And let me just give you some scenarios here that are very, very important for you to remember. You'll find some cases where growth may slightly increase. You see, look at the monocotyledonous plants. Monocotyledonous plants will have their growth increasing slightly until the organism dies. So there's increase in growth. Increase in growth until the organism dies. So that means monocotyledonous plants like maize, sorghum, millet, rice, you see, and grass will have their growth increasing slightly until they end up dying. So in other words, their growth does not stop. The growth proceeds at a very slow rate, slightly, until the organism ends up dying. You see, and many invertebrates like the insects, the crabs, and the class Crustacea, or the wood lice, or the Daphnia, and the chilopods, the diplopods, that's actually the centipedes and the millipedes, and most other organisms like the ticks, the mites, the spiders will actually end up also having their growth increasing until they end up dying. So they might have the same experience as the monocotyledonous plants. Even the fish, you see, as vertebrates, will also have their growth increasing slightly until they end up dying. Look at some reptiles, like the crocodiles, you see, the chameleons, the lizards, you see, those organisms, and even the tortoises will have their growth increasing slightly until they end up dying. Look at that. So that means the growth increases slightly until the organism ends up dying when it reaches its maturity. Now, in some other cases, you'll find growth, you see, increasing until the curve ends up flattening to show that there's no any further increase in growth. You see, and in some cases, the growth curve may tail off. Look at this. So I've said in monocotyledonous plants, some reptiles, some invertebrates, and some fish, the growth curve slightly increases. Increasing means showing an indication that growth is slightly increasing up to a particular point where the curves may actually show that the organism has died. The curve increases slightly, growth increases slightly until the organism reaches its maturity. Then it later on dies. Look at that. But in some other cases, you'll find the growth curve increasing, then flattening out. The growth curve increases, then flattens out. Look at that. The growth curve increases, then flattens out. In some other cases, you'll have a curve whereby the growth curve will actually increase, then it will actually tail off. Look at that. So there's a difference between flattening out and tailing off. You see? So we'll literally say that there are three main types of growth curves. One, a growth curve where there's increase in growth. Look at that. There's no flattening out, there's no tailing off. Growth increases until the organism dies. For example, the monocots, the reptiles, the invertebrates like the insects and arachnids, and then the fish. Look at that. Their growth increases until they die. In some other organisms, the growth increases, then the curve flattens out to show that growth has stopped or ceased. This is what we call the plateau or the stationary phase. In some other organisms, growth increases slightly, and then at the end of the day, it actually ends up having its growth curve flattening out. So this negative pattern of growth, where growth increases, then the curve flattens out or the curve tails off, can be seen in mammals. So mammals have this particular type of growth, where they increase in growth up to a particular point where the curve flattens out or tails off. For example, in humans, you'll actually be able to experience this. And in this case, that means there's what you call physical senescence. The cells have ceased increasing in size and in number. Physical senescence is associated with increased age, where there's no any further increase in cells. In fact, the number of cells dividing is less than the number of cells dying. Look at that. The rate of cell division is either equal to the rate of cell death, or the rate of cell division is lower than the rate of cell death. And you see, sometimes they can just ask you to explain any three types of graphs that can show there's growth or there's no growth, or they've actually been told to explain how growth occurs. We'll actually talk about one, the growth rate increasing until the organism dies, or the growth rate increasing, then the graph levels off or flattens, or the growth rate increases up to a particular point, then the graph tails off. I've talked about growth rate increasing until the organism dies in monocots, reptiles, invertebrates, and fish. I've talked about growth rate increasing, then flattening out or tailing off in mammals such as humans, where there's what we call physical senescence. There's no any further increase in size of the body, and that is actually associated with increased age when human beings grow. Now, a sigmoid curve does not apply to all organisms. I'll not talk about sigmoid curve in arthropods or invertebrates like insects. A sigmoid curve will apply in most cases to some invertebrates with an exclusion of the insects, and even some more invertebrates that have what you call a cuticle that normally gets shed through molting or ecdysis. And of course, vertebrates will not have the same aspect of what you call a sigmoid curve. So that at the end of the day, if someone talks about a different type of curve that brings about growth, you won't talk about the sigmoid curve. We'll talk about a different curve. So a sigmoid curve does not apply to arthropods, for example, such as insects, where growth occurs at intervals. So insects have growth occurring at intervals. Growth occurs at intervals. Growth occurs at intervals so that the volume can change, and as the volume changes with the time, you can actually plot those two parameters. And what you get is not a sigmoid curve, but an intermittent curve. Intermittent curve. So when growth in insects is monitored, then that means we will end up plotting, for example, volume of the insect or the grasshopper against time. And that means we shall get what we call an intermittent growth curve. Intermittent growth curve. Intermittent growth curve. Intermittent growth curve. So we shall call this an intermittent growth curve. Slightly different from what we've actually been discussing, and you might actually be able to get confused thinking that the growth curve is the same. No. So the intermittent growth curve can be of this type. For example, let me just make a sketch. Then I can put here time. Then I put here maybe time in seconds or in hours or in minutes. We can talk about, let's talk about time in hours. That's better. Time in hours. Then you can have volume. Of course, a volume is a parameter that can be used to measure growth in insects. Or you can even talk about length. And it can work for us, length in centimeters. So you'll have something of this sort. And you can have this. Yeah. So at this point, there's what we call molting. Molting is a process where the exoskeleton, because arthropods have their skeleton to the outside, so their skeleton is called an exoskeleton. It is made up of chitin that is shed or actually secreted by the epidermis. It hardens on getting secreted, so it actually forms the backbone, or actually the skeleton of these animals, protecting the internal delicate structures of these organisms. So there's what we call molting or ecdysis. Look at that. So that you shall have what we call the first instar or first growth, what we call second instar, the third instar or growth, what we call the fourth instar or growth, what we call the fifth instar or growth. So first instar, second instar, third instar, fourth instar, fifth instar of growth. So at this point, there's growth. There's growth. Look at this. Let me give you an example. Here, there's molting or ecdysis, where the exoskeleton gets shed. When the skeleton, the outside of this body's arrangement is shed, that means we shall follow this process by what we call growth. So again, the skeleton again forms, it is again shed. After getting shed, then there's again growth. So one, molting or ecdysis to remove the skeleton, then there's growth. You look at that. Molting, then growth. Molting, the skeleton forms, shedding, growth. So I expect to have first the skeleton getting formed. You see, the epidermis secretes the skeleton, it forms, it actually covers the body of this organism. At this point, the skeleton gets shed if the organism wants to grow. Then there's growth. Look at that. At this point, growth has occurred. The skeleton forms again. It is shed, the organism grows, the skeleton is again formed. So, first instar or first growth, there is molting to remove the skeleton. The skeleton again forms, the skeleton is again shed, there's growth again, the skeleton again forms, there's shedding of the skeleton, growth is occurring also. So that's how it occurs. Now, during intermolt phase, that means intermolt phase to mean when the molting has stopped and the skeleton is again forming. So we have what we call molting, then molting. So molting at this point, molting at this point. Between these two, intermolt phase. Look at that. And that literally tells you that now the tissues are expanding. The tissues are increasing in size. So this is an intermittent growth. Intermittent growth occurring in arthropods such as insects, which have their skeleton to the outside, called an exoskeleton made of chitin that is secreted by the epidermis. We've said first there is shedding of the skeleton through molting or ecdysis. Then there's growth, for example, the first instar. Again, at this point, the skeleton has to form. You see, when the skeleton forms, it is again shed. Then the organism again increases in size. Again, the skeleton forms, it is shed, the organism again increases in size. That's just a good summary of how you can be able to explain the process of growth. But remember, still during the intermolt phase, there's still growth occurring, there's expansion of tissues and cells, and the organism can still be able to actually get seen increasing in size or volume.